Hi, everyone. Thank you for being here with us today. We are watching group 2, which is environment. The teams are going to present, the judges are going to ask the team some questions. And here I wanted the judges to present themselves really briefly with their names, institutions, and country. Judges, please.
Okay, then I'll go first. Hi, my name is Christian. I'm from Germany and I work at the Center for Biotechnology in Bielefeld.
Hi, my name is Luis. I'm currently a test engineer at Ginkgo Bioworks in Boston, Massachusetts.
Hi, I'm Matsuri Rojano. I'm a PhD candidate in Austin, and I'm originally from Mexico.
Hi, my name is Yusuf. I'm from Egypt, and currently I work as a biology content developer at Nagua Technologies.
Marco, please introduce yourself.
I think that he couldn't. Okay.
You're on mute.
Hi guys, can you hear me? Sorry. Well, I'm Marco Pereira, I'm from Mexico. I'm PhD in molecular biology. And well, I'm studying now, I'm professor teacher in different universities in Mexico.
Great. I thank you all so much for being here today, judging the groups. I'm going to let the first group in, which is UNB in Brava.
Hello.
Hi.
Hi there. All right, now that everyone joined, we're going to start the session. Just a reminder, you have 20 minutes. I'm going to let you know when you pass 10 minutes and then 5. And Verônica is here to help with the translation. Vou falar em português rapidamente. A gente tem 20 minutos, então eu vou avisar quando passar os 10 e depois quando tiver 5. A Verônica está aqui para ajudar com a tradução. All right, judges, the floor is yours.
The first question, Christian, someone?
Yeah, okay, then I'll go first. Let me start with saying that it's a really nice project. We worked in a team with microbial fuel cells almost 10 years ago, so I really like the concept. But I have one question concerning your kill switch. I see that you really like the concept of a light-induced kill switch, but do you envision any problems using it with a fungus, which can grow in the dark? So how do you ensure that the fungus is really killed if it's released in the environment?
Vocês entenderam a pergunta? Alguém se propõe a responder em inglês? Lucas?
Eu posso responder em inglês.
Okay, perfect.
Well, I'd like to start off by thanking everyone for being here, but especially about the kill switch. We envisioned and we based our system on a previous project of another team, of another hygiene team, and they used a sort of membrane, but like a film, not a membrane, a film that would allow only a certain wavelength to pass through it. So we would ensure the chamber of the microbial fuel cell would only have a certain wavelength of light inside of it. And then if the fungus would eventually escape or go out of the MFC, it would be killed by the specific red wavelength of light. So yeah, did I answer the question?
Yeah. What I'm so how do you ensure that the fungus actually sees enough light? I mean, we are talking about a lot of water there, which is pretty dark. So light would not be the first thing that comes to my mind if I want to kill something that escapes from my container.
Well, the container itself would be contained. It would be closed. So ideally, the fungus and anything else would be contained in the container. So the only possible event where the fungus would be exposed to the light is if it would be exposed to outside of the container. And if that happens, it would be the water, the leachate would be in a flat surface. So the fungus would be exposed. I imagine like physically it would be exposed. And I think your question is regarding like if we would have biofilm of some sort, probably the inner layers of the biofilm wouldn't be exposed to the light, but that wouldn't be the case if we are talking about an escape of the containment, because the water will be flat. And ideally, after the treatment with the bacteria, the leachate would not be as dark anymore, because we have studied and we have researched that bacteria, especially the Geobacter sulfurreducens, also helps with the color aspect of the fungus of the leachate, so it would not be as dark as it was when it first came to the MFC.
Okay, thanks.
I would like to make an observation. That is, our lacase is also able to reduce the color of leachate, and our project aim to be implemented in a certain part of the treatment in ASB, the sanitary landfill. So in that part specifically, the leachate would be pre-treated in some way and will be, when exposed, it would be more light, the leachate.
Clear.
So I think clear. It isn't such a big problem, I think.
Okay, thanks.
Okay, I think I can go. First of all, I liked your project and especially the presentation video. Before I ask my question, I'm just curious to know whether you like recorded it in a different language and then added the English voiceover as voiceover, right? Or was this the case? Okay. Okay, for my question, I want you to elaborate more on the choices regarding the cathode. Like, why went with the bigger cathode and what other choices and why you didn't choose any of them? Like you mentioned some of them, but I want you to share with us.
Did you understand?
Yeah, I think Julia can.
No, I didn't understand.
Ele perguntou sobre a escolha do cátodo e sobre o tamanho especificamente do cátodo, por que a gente fez esse tipo de escolha.
Oh, okay. I'm gonna try to explain. So the cathode we choose to be bigger than the anode, so it could increase the efficiency of the MFC. And in the cathode we insert an air pump, so it could increase the difference of potential. And we also choose a carbon cathode to be more poroso. I don't know how to say poroso. So it could be better to the fungus. So it's this.
In our choice, we talked to some specialists, and they told us that the cathode was the limiting part of the MSC. So that's why we choose like a bigger cathode.
But was there other options that you choose not to go with, like another way to solve this problem of the cathode being a limiting factor?
The cathode being a limiting factor is that, well, we also put the cathode on the upper area, so the flow could be upper, and we saw that the cathode being upper could be better to our project and make the efficient better.
Okay, thank you. Thank you.
I have a question next, if that's okay. So congratulations on your project, first of all. I know that you've, like, obviously in this format of the competition, you focus a lot on the design of your project. And you listed like a lot of your parts come from like different organisms. So as part of your design process, what have you considered like to make sure that all of these parts will express on your chassis? And do you think there's gonna be some like codon optimization that needs to take place, or what strategies are you considering?
First of all, I'd like to address that we actually envisioned doing codon optimization in the first place, but we didn't because this would mean that we would have to register every new part as a new biobrick. So we would be using other biobricks, but because of the codon optimization, we would have to register them as new biobricks, as new parts. So that was a little counterproductive at the time because we didn't have like really the time to do it. But we do envision that in the process, and when we go to lab experiments and when we try to do it for real, we're going to do codon optimization for sure. Definitely. We also try to always implement every part being from similar organisms. So we try to find our biobricks primarily on fungi, and then other eukaryotes before trying to go to bacteria or stuff like that. So we try to go that way before. I don't know if that makes sense, but we learned about that in Aging Academy actually. So I think that was our main concern when building the circuit.
Yeah, that answers my question. Thank you.
Thank you.
I guess I'll go next. I'll just reiterate what the previous judges have said.
Just a second, you have 10 minutes.
Ooh, all right. You guys did fantastic. Love the project, love the visuals, love the presentation and for your human practices. I think you guys focused on, I believe, the deaf community, which I thought was unique and really, really cool. My question to you is a technical question. Have you thought of how much energy you can generate in your MFCs and like how much leachate it would require to generate that energy?
I would like to answer first, and I think that if someone needs to complement, it will be nice. But we have a talk with a professor called Punch, and he helped us with a table that have when we part to the experimentation in next year that we aim to do, we can calculate through this table. So in Design League, we couldn't do this because we haven't the experimental part, but we have a lot of studies in the literature and the papers to think in the best way to produce energy in the next year, we can also test this physically, I hope.
I'd like to just add something to it, because first of all, we actually did a lot of research on choosing the microorganism to choose the most efficient organism that could generate more electrons extracellularly. So Geobacter sulfurreducens is one of the best bacteria to do so. And this doctor that we talked to, Dr. Deepak Pant, he gave us a spreadsheet. She was trying to, it's stable, but it's a spreadsheet. It's an open access spreadsheet that we got from him, and we can input everything about a project there, and it gives us some parameters about electricity generation efficiency and stuff like that. But we could not do it in our project like in a very efficient way, because we couldn't do practical work.
We didn't know the specific like components of the leachate.
Yeah.
To go.
So in our project, for example, we use a lacase that can degrade a lot of components. It has a lot of substrates, but we would have to choose a specific one to generate such a result. So we didn't do this specifically yet, but we do intend to do it in the future. So
yeah, furthermore, to calculate the efficiency of the MFC, we have to make analysis of the demand of oxygen, and then when we have these values, then we will be able to say that the efficiency of the MFC based on this table that Dr. Pant told us.
Thank you.
Any more questions? We have 5 minutes.
Yeah, then maybe to clarify that in your MFC design, it looks like it's a continuous flow. But if I now understood you correctly, it's basically just to fill the MFC and then you let it run for some time and then you basically flush it. Or is it a continuous system?
I think there are people that are more qualified to answer that, but I'd like to start saying that we envision, first of all, a batch system. So there would be a batch, and then the reactions would take place in that batch, and then it would be released into like the next step of the treatment. So it would be a batch system, but it would also be continuous, because this would be happening all the time, like with no stop. So I guess it's a batch system, if that's the question, but it would also be continuous.
The reason why I ask is that your design basically assumes that there is a time when the lacase can bind all the copper, and afterwards it switches. If it's a continuous system, you would always flow out the lacase and flow in new copper, so your system would never trigger.
Exactly. Okay. So that's why we would have a batch system in which basically it would fill the container, and then the flow would stop, and then every reaction would take place, and then the circuit would work its way, and then after everything happened the way it's supposed to be, then we flush it out, and then more comes in. So that's the idea.
Okay, thanks.
Thank you.
Anyone else? We have time for one last question.
I'll ask one. Have you guys thought of the long-term stability of your circuits once they're deployed in these organisms? Like, let's say you build them, they work great, you have them growing in your MFCs. What happens if, like, after 2 generations, your circuit breaks and things that aren't actually functional overtake the population? Have you thought of ways to kind of select for things that still work?
Yeah, we actually, for the integration of the circuit to the genome, well, first of all, we're trying to integrate it to the genome. So we research regions and I'm sorry, we research papers that actually described regions of the genome of the Aspergillus flavus and other Aspergillus fungi that would not be essential to the organism. So we aim to integrate these parts into non-essential parts of the genome. So there will be minimal like compromising of the metabolic activity. And if something happens and we still want to salvage something that will work, there are separate selection markers to each plasmid. So we use 3 plasmids to accommodate everything that we wanted to put in the organism. And each of them have different selection markers. So we can, okay, so like this part is not working anymore. So we can kill all the organism that don't have this part with this antibiotic, but we use this other antibiotic to select the organism that still have something that works. So we use different selection markers for that.
Thank you.
All right, thank you, team. Thank you, judges. Your time is up. So I'm going to ask you to please leave the room. Congratulations on your answers. And if you guys want to, you can keep on watching the other teams on YouTube, okay? So thank you very much.
Great job, guys.
Thank you very much.
Thank you.
Thank you very much.
Thank you. Bye.
I'm going to ask everyone who's in this room to change their name to their name of the team and underline your name, because that would help me a lot with the letting in or out who's who. And I'm going to gently ask those who are not in the next group, the next team, to please leave the room and come back again whenever it's your turn. So we're going to start with IS Panama right now.
Just a quick message. Thank you, Chris. We're still having problems with the streaming of this specific room. Don't worry. If we keep having problems, we can stream it not live, but after the judging session is over. And well, don't worry for the hour. We will respect your 20-minute time, so probably a bit with a slight delay. So please go ahead and wish you the best luck in this judging session, Team Panama.
Okay, so now we're judging IS Panama, and feel free to start.
Okay, then I'll go first. So let's start with, I really liked your project proposal. Getting rid of pesticides, I think, would be truly an achievement if that can be realized. And that brings me directly to my first question. Why did you choose to enhance alcohol production? Is that really the rate limiting step with the degradation of organophosphates? Or wouldn't it be better to express enzymes that actually attack the organophosphorus compounds?
So hello, first, good afternoon. We decided to use alcohol, specifically ethanol, because so we did like research and we saw that there was information that our specific chassis of the organism was able to produce ethanol at a very slow rate. So our idea was just basically to overexpress a pathway we selected and basically degrade the pesticide into ethanol. That ethanol can be used as a biofuel. And so we saw that it was an easy and concise way of having a good transformation of the pesticide into ethanol that is a compound that can be that is biodegradable and has a neutral pH. So it was like a better outcome in that way we saw.
Thanks.
Hello, team.
Hello.
Oh,
Oops, sorry.
you can go, Luis.
I apologize. Same as Christian said, love the project, well executed, liked it a lot. I have just a general high level question in your design. Do you know if the organism that you're trying to engineer to degrade these compounds requires transporters to get those compounds like into the cells in the first place?
Can you repeat the question, please?
Do your engineered organisms require transporters to import the organophosphate molecules like into the cells to do the degradation?
We don't believe so. Basically, what we focused is that the organism by nature was able to transport the pesticides in a natural way. So we didn't have to add any artificial parts to basically eat for the organism to eat the compound. So it was like natural. So we only overexpress the metabolism to degrade the pesticide faster.
And I guess a follow-up question, do you have a sense of how sensitive these organisms are to the organophosphates? Like if the organisms were deployed in an environment that was like really, really high concentrations, would they just die? Could they actually survive and degrade things? Do you have a sense?
Well, I'm not sure, but I think what we found out is that basically the degradation goes from in terms of like surviving the organism in a volume with high pesticides. Basically, the metabolism, the degradation is going to go from a very acidic pH level to a basic level, and so the organism is able to survive that level of change of pH. And also, well, the objective of the project was to degrade pesticides in an environment where the amount of the pesticide is mixed with another chemical or compound. It can be either soil, it can be water. So probably we cannot use the organism in environments with a lot of pesticides in the content, but yes, with a fair amount of pesticides. Still, we need to research that specific amount, like the threshold limit. So it's a good question for future implementations.
Thank you.
Thank you. So I guess I will ask my question next. So I have a question on a slightly different area of your project. So you mentioned that you as like your product being a powder. How do you decided on that format, like having it be a powder versus other things, and what kind of what were the experts or like people that you contacted throughout your project? I think it will be really cool if you can talk to us a little more about like the different interactions that you had with stakeholders.
Okay, so first of all, I think we mostly contacted one of our mentors, who is Edina Fuentes, and he's one of like the main maybe supervisors of this iGEM in like our team. And he talked to us a lot about what we had to keep in mind when we made this product, the powder product, basically how we were going to make it so that it's still sustainable, because we obviously can't just put any powder into any system for it not to be sustainable. So we had to take those things into account. And that led to a lot of individual research, I would say. But I think most of what we researched about the powder product was the entrepreneurship side of it. And I think maybe another one of my team members who's more knowledgeable about that part maybe can speak up on that. But we talked a lot about, we researched a lot about what the economic aspects could be and the implications of that in our project.
Yeah, so essentially we decided to do a powder format since for our vision for our business side of the project would be to select different quantities of the bacteria to be located on the different natural waterways. So therefore, different powders allow easier transportation of the bacteria, and it allows easier like we can like transport the bacteria easier and like give it to like the consumer in a more easier format. So that's why we set up the powder format than other systems.
Yeah, also adding up what my co-teammates mentioned, in terms of the biological aspect, we decided to use powder in the form of powder, mainly in the side of what Judge Luis mentioned, that we can add specific enzymes or nutrients to the organism in the powder to make sure it does the degradation in an efficient way, and we know for sure that the organism is going to degrade. Just adding specific enzymes or nutrients just to make sure the organism doesn't die in maybe in a volume of like a high amount of pesticides.
Okay, thank you, Tim.
Actually, I loved your project. I especially loved how diverse is your team. So good for that. I want to ask you about something you've mentioned. You stated that the agriculture sector in Panama is known for using like pesticides more than other countries. Can you explain that? Why do you think this is the case in Panama?
Okay, so in a very general sense, Panama relies a lot of agriculture, and most of the rural sector and the population that lives in poverty relies a lot on agriculture. But since Panama doesn't have many sustainable educational campaigns, there isn't a lot of talk about how we can have more sustainable farming methods. So methods of farming are very traditional here and rely a lot on the pesticide usage. Panama imports actually a lot of pesticides a year, and it has had like a very big increase in imports of pesticides because these traditional methods are just continual and they continue to be used. So that is one of the main reasons why we believe that Panama still uses a high amount of pesticides. And it's another reason why we believe our project could have such a big reach, because beyond being just the biological aspect and the solution aspect of it, we also believe that it's a very good thing to have awareness and like create an educational campaign on this so that people are more aware of the impacts of their farming.
Yeah, great. Another question I want to ask you, you are talking about when you start testing your system, you want to test it on different chassis. And you mentioned E. coli as one of those chassis. Have you considered any other chassis to test your system in?
Before you answer that, I just wanted to say you have 10 minutes.
We basically selected those two main chassis as our of the development of the idea was based on the cyanobacteria organism, but in terms of the actual creation of the organism in a lab environment, we decided to use E. coli because it was much easier to construct the organism in a chassis such as E. coli. But we only selected those two organisms as we didn't want to work on many organisms, as we wanted to make sure that our focus on the project stayed on the cyanobacteria, but using E. coli as application part of the organism.
Okay, thank you.
If I may ask another question concerning your kill switch. So if I got it right, that is basically triggered by a shift in the pH. But your system is used in the soil, right? So do you envision that the degradation of the organophosphorus compounds changes the soil pH that much that the kill switch is actually triggered?
Thank you for your question. So basically, at first, we were going to use a light regulated system, and that we mentioned that on the improvement of previous IGEM projects, because we saw that previous work integrated a light regulated system. But the thing with light is that the amount of time that it takes to degrade like a dynamic amount of pesticides, it was not fixed. So we decided to use a pH system, as we were just going over the project with Edina, one of our mentors for the project, and he mentioned that we can take a look at the pH system. So then we did more research, and we saw that I don't remember the specific title of the paper that researched the pH change. But the results of the investigation just basically mentioned that with medium to high amounts of pesticides in soil, the change of pH is noticeable. So based on the results for that investigation, we saw that a good pH level to detect the change was going to be around 5 to 6. So we selected a pH detector of a pH of 5.5, and that is mentioned in the document. So that was going to be our threshold to detect the change of the pH.
Okay, thanks.
I'll ask another question if there's time. So in your report, you mentioned that you at least list the cost of like a septic system as an alternative that could be used to kind of like filter out some of these compounds out of the environment. And you mentioned that your engineered organisms could be a more affordable alternative. My question to you is, did you factor in the potential like research and development costs that would go into engineering that organism to the point where it could be like commercially viable? Do you have like a feeling for what that might cost?
We didn't factor that in, but we believe that if this process like grows large enough, it could potentially like the marginal increase of the next bacteria would like be heavily decreased. So like if this expands large enough, if we sell them to several government organizations or like nonprofits, like Solia Water or Disgas, which is Panama, the wastewater treatment of Panama, we could believe that the marginal product increase would offset the high research costs that might confront.
Understood. Thank you.
Adding on what Marcelo said, just like a quick comment. We didn't maybe factor the cost of development in the overall entrepreneurship part of the project, because the development was going to be like a one-time cost. Maybe like maintenance would require funding to improve and maintain the project. But at first, we evaluated that the cost of the developing side of the project wasn't going to be in a scale compared to the amount of money that is needed to fund traditional methods to remove pesticides. We saw that overall the cost of our project was going to be much lower. So we think that maybe the value that is going to cost the development wasn't going to be like a lot compared to the positive aspect economically of the project. So.
Thank you.
We have 5 minutes. Does anyone else? Oh, I'm going to let you answer that, but if you guys have another question, we have time for one more.
Yeah, I just wanted to add on a little quick thing about the economic cost of it. We also believe that, keeping in mind all of the things that my two teammates have already mentioned, the economic benefit of having a more sustainable farming method for a community is very great, because as we mentioned in our project page, pesticide poisoning is a problem that affects many communities here in Panama and has lots of fatalities and affects the economic success and the economic growth of communities. So we believe that with the implementation of this project leading to healthier and more labor enhanced communities will also make the economic status of the country and the communities increase. So it's a double benefit to that too.
I could add one more question if we have time. So I know that initially you're trying to maximize the production of these enzymes, but have you considered like any metabolic impact of like having like always producing that enzyme? And do you think that in the future, have you considered implementing like any control systems on your design?
Well, at first we focused on preventing GMO contamination. So our idea was, when we add the organism to remove pesticides in soil or in the medium we choose, we didn't want to contaminate it with a modified organism. So our main idea was to use a self-destruction kit based on the pH detector. And we didn't focus a lot on like any extra prevention of contamination, as the chassis and the organism that we use is like very safe, especially for humans, and so it doesn't have any side effects. And in terms of overexpressing the cycle that we mentioned in the document, we made sure to research that it wasn't going to have any secondary effect on maybe for humans or for the environment in general. So it's in theory the project should be very safe in terms of contamination and the environment.
Yeah, just to clarify my question, sorry if I got in the way, I wasn't clear. I was thinking more on the on your strain, on your chassis, like if there was going to be an impact on its metabolism or like its ability to survive and replicate, just for like always producing this enzyme and focusing a lot of its energy on that specific process.
Well, for the organism, basically what we're doing is that we are only overexpressing the enzymes that are already in the organism. So we are not adding any extra enzymes to the organism. We only selected a series of enzymes that we could overexpress, and the overexpression should not have any effects on other parts of the organism. The organism, it will be just cyanobacteria focused on degrading a pesticide. But for future experimental process of the project, we are going to take a look on the side effects of maybe the life length or maybe the replication process. But at first, I think we believe that this cyanobacteria shouldn't have any problems with the overexpression of the enzymes. So I think that answers the question.
Yeah, thank you.
Thank you so much, judges and team. Congratulations on your answers. This is the end of your session. So please, if you guys could leave the room and watch the rest of the teams on YouTube, that would be great. And now we have UTFPR, UT primers. I'm going to let them in.
Thank you, bye.
Okay, so hi everyone. UTFT, sorry, UPFT, UT primers, it's easier. You're here now, so I'm going to start your time. And you have 20 minutes. I'm going to let you know when you guys have 10 and then 5, okay? So judges, the floor is yours.
Okay, I think I'll go first again. And for the one reason that your project I really like, because you're trying to do something that I've done myself in my career, so making carotenoids. So I like your design, but I have one question slash suggestion. You start by introducing the MapDocs pathway. Have you looked into other pathways that can achieve the production of IP and DMAPP that you need?
Okay. They are Portuguese speakers, right, Cris? Alguém de vocês se sente seguro para responder em inglês? Ou vocês entenderam de fato a pergunta do Christian?
Se conseguir repetir, eu acho melhor para a gente a pergunta.
Yeah. Christian, could you repeat, please? So I will also pay attention on your question.
Se conseguir traduzir para a gente também daí?
Perfect. Okay,
Sure,
I'm
sure.
going to. Okay.
So I really like your project because I worked with carotenoids. Concerning the first circuit you want to introduce to make IPP, have you considered other circuits?
Relacionado ao processo para fazer IPP, I don't know exactly what is IPP, but se vocês consideraram outras moléculas ou células para fazer esse processo. I don't know if I'm totally sure of this, but tem sentido essa frase para vocês em português? Essa pergunta?
Ele perguntou sobre o primeiro circuito. Não sei se
eu acredito sobre a via metabólica, né?
Acho que sim.
Por que a gente não escolheu outra via metabólica? Eu acho que é isso.
Isso mesmo.
Então, primeiramente, a via metabólica do MEP que nós escolhemos, ela já está presente naturalmente na E. coli. Isso foi um ponto que a gente decidiu, né, escolher essa via. Mas...
Ok, vamos devagarzinho, okay? Para você me ajudar a falar com ele. Então, the first motivation to choose this one is que a E. coli ela é presente
a via
naturalmente.
MEP já está naturalmente na E. coli.
Yeah, okay.
The organism E. coli, that much I got.
It's naturally present on this on E. coli. Did you got it? Like it's naturally present on this.
Yeah.
Yeah. Go ahead.
Tem mais um argumento que eu queria só para complementar.
Claro.
E a via MEP era mais energeticamente equilibrada e ela tem um maior rendimento de conversão de massa de glicose para a produção de terpenoides.
Ok, essa via MEP ela é energetically more stable. I don't know if it's clear, but this EMP is energetically more stable than others. Ela é mais estável que outras vias. É isso, Gabriel?
Ela é mais energeticamente equilibrada e tem maior rendimento de conversão de massa de substrato em terpenoides.
Wow, this is hard for me.
I think I got it at least in part. Yeah. So then just one suggestion concerning that. I'll put it in the chat. Actually, you might want to look into this paper because that is a pathway that contains only 2 enzymes. So it's very short and it's very easy to force the production of IPP with it. So if you really want to have a high yield, use that one.
Are you suggesting something that is in this paper to them?
That they can use.
Ok, ele está sugerindo para vocês, ele enviou aqui, não sei se vocês já acessaram o link, e aí ele está sugerindo que vocês, óbvio, leia e estude esse material que tem um método, uma forma que pode ser mais interessante para vocês, ok? Eu acredito que esteja relacionado a essa pergunta dele. Ok? Vocês pegaram direitinho, guardaram o link? Tranquilo?
Ok, obrigado.
That's nice. Any other question, Christian?
I let the others also ask something.
Okay, I think I can ask a question. The compound, the oxybenzone, this should be the oxybenzone, is you are talking about it as a potential risk and a cause for many diseases. But in your search, have you found any data that suggests that it's safe?
A pergunta seria no sentido que ele está falando do óxido benzoílico, eu acredito que pela proximidade seria isso. E ele falou assim, se nos dados que vocês encontraram, vocês têm alguma referência se ele if it is safe. It's it?
Yeah, I want to ask if they found something that says if it's safe, like what?
Okay.
Yeah, I want to know if they like did the search about this.
Isso. Então ele quer que se vocês fizeram alguma pesquisa e identificaram algum dado que fala que é seguro, que it's safe to use this, utilizar isso.
Ele está perguntando se a oxibenzona, se mesmo que a gente tenha achado que ela é ruim, se todos os nossos dados também acharam alguma coisa que ela era boa. Tipo, se teve alguma contradição nesse sentido, sabe? Acho que o Peterson pode responder bem essa parte. A gente não está te ouvindo, Peterson.
Yeah.
Não sei por quê.
Bom, eu posso iniciar tentando responder essa pergunta e se o Peterson achar interessante, ele pode tentar complementar. Mas dentro das nossas pesquisas, né, a gente encontrou vários artigos que apontavam principalmente para essa questão do branqueamento dos corais. Porém, uma das pessoas que nós conseguimos conversar, né, que é a Mariana Bata Alonso, que é uma pesquisadora e ativista dos contaminantes marinhos, ela nos deu, né, esse contexto histórico de que esses protetores solares à base de oxibenzona, octoprileno, eles surgiram, né, num contexto de tentar, de alguma forma, substituir os protetores minerais que ocorriam na década de 80 e 90, que produziam uma pigmentação mais forte, mais esbranquiçada, e que tinham um potencial maior de reações alérgicas. Então, houve um processo de aceitação pela Anvisa, porém, como teve um processo de aumento do turismo marinho nas últimas décadas, né, esse composto ele começou a ser acusado dentro de tecidos de animais marinhos e também como responsável pela morte dessas plantas.
Vamos ver se eu consegui pegar tecnicamente. Então, seria a diferença aí entre esse composto é um composto mineral, o que contrapõe outros compostos que era dióxido benzoico.
Bom, a oxibenzona ela veio para substituir, né, esses compostos
minerais. É o contrário, então. Então tá. O composto que é o foco de vocês, ela vem substituir os protetores que eram minerais. Certo?
Eu sei mais ou menos inglês, eu acho que eu entendi a resposta da Evelyn. Você quer que eu fale direto? Que aí, tanto que eu já tô a par da pesquisa, né?
Yeah, because for me, technically, it's hard to
I understand. Historically speaking, the oxybenzone and also the octocrylene that we researched that we did the research, they came after a mineral compound that they used to use in the sunscreens because they were worst, and they started replacing it because they thought it was better, but then the amount of people using it increased as well as the world population, so many people more were using it and it went to the seas. So that's why they started seeing these compounds into the tissues of animals and other reefs, coral reefs, for example.
And Larissa also said something about if it is for the animals, it is safe for the animals, or is it not make sense in this question?
I'm asking if they found any data that suggest the opposite, that suggests that.
Okay.
Yeah.
We found only about the bad parts of it, because, well, the only good part was the fact that they were replacing and the population actually accepted that, because the ones that came before were much more worst for our skin and etc. So that was the good part, but making more research, we realized it's not good.
We also talked to a professor.
Just a second, Larissa. You have 8.5 minutes.
Okay. So we also talked with a professor that researches this kind of thing. And for her, it was, she found that the oxybenzone was really bad for the animals as well. For the dolphins, I think, more specifically, because they are natural kind of finders of pollution and things like that in the ocean. So
one last question. How the current oxybenzone-free sunscreens work? Have you looked into that?
Gente, eles perguntaram como os protetores que não têm oxibenzona funcionam. Qual é a diferença entre eles, basicamente. So basically, our proposal is to make a sunscreen with carotenoids that is better for the environment, right? So the difference between... do you want to... Okay. So basically, the oxybenzone is really bad for the environment, and our proposal is to make a sunscreen that uses only organic molecules, which means carotenoids. So when they go to the sea, there's no damaging residue, basically, because carotenoids can kind of disintegrate very easily, so it wouldn't leave any residue. Basically, that's that.
Thank you.
Any other questions? We have 6 minutes.
I'll ask a question. I'll start by saying I absolutely love this project, but like the amount of detail that you went into in your report, going into how you would formulate the new sunscreen, like the visuals, the presentation, everything else, it's like it was great. It was great. My question to you is, why use E. coli as a chassis? And have you thought about using other organisms as chassis to produce these compounds?
Ele perguntou se como a gente chegou na questão de usar E. coli como chassi e também se a gente pensou em usar outros chassis. So, alguém quer responder? Eu posso começar a falar que a gente tinha pensado em usar outros. Tá.
Quer responder, Gabriel? Quer que eu assuma? Aí se a Lari puder traduzir.
Eu posso responder se quiser.
Responde, então. Acho que fica melhor.
Então, a E. coli, ela não produz carotenoides naturalmente, mas nós optamos por obter ela como chassi, porque ela é um organismo modelo, né? E há muitos estudos sobre a produção de carotenoides nela. E além de ela já realizar a produção naturalmente de isoprenoides. Não sei se quer traduzir isso já, Lari, ou posso falar tudo?
So basically, E. coli is a natural model. So we can study it better. It has more options of articles and things that we can consult, basically. And E. coli also has a natural route of isoterpenoids. Eu acho que é isso, Gabriel. Eu falei que a E. coli tem uma rota natural de isoterpenos e que tem muito mais estudos sobre a E. coli. Tem mais algum?
E a gente conhece apenas um micro-organismo que produz o carotenoide que a gente precisa, que é a Rhodobacter viridis, da cepa JA737, só que ela foi descoberta recentemente e por isso a gente decidiu não usá-la, né? Porque tem poucos estudos em cima dela e a E. coli facilitaria muito mais o nosso projeto.
So basically we had another bacteria that also produces... o que essa outra bactéria produz? Perdão.
Neurosporina.
Okay. This bacteria, the I don't know.
Rhodobacter viridis.
Rhodobacter viridis. It produces the Neurosporina. don't remember the name in English. But it basically produces this, but it doesn't have so much study. So we don't choose E. coli because it has more studies about it. So it would be more, we could do a more complete work, and we had more data for this work.
Thank you.
Maybe a short comment to that. Stay away from Rhodobacters. They are notoriously difficult to work with.
Yeah, that's one of the reasons that we didn't want to use it. It would be more difficult as well. And E. coli is better, is more resistant to work with, so it would be easier.
All right, thank you, team. Thank you, judges. Your time is up. Congratulations on your answers, and please leave the room and watch the other teams on YouTube. We are now going to have a break of 5 minutes. So we'll be back, well, it depends on the time zone you're in, but in 5 minutes, okay?
Nice job, guys.
The judges for your input. It was very nice to be part of it.
Lembrem do artigo da sugestão do Christian.
Sim, já guardei o link e vou passar para o grupo.
I'll put something more in my comments to your team. Also, people to contact, actually.
Oh, perfect. Thank you.
Thank you, Christian. Bye-bye.
Bye-bye.
Great time. Bye-bye.
Thank you. Bye-bye.
Thank you.
Thank you. Bye-bye.
Bê, não sei se você viu minha mensagem, mas esses já foram os times do Brasil, então você está liberada se quiser.
Ah, tranquilo. Os outros meninos são de quais países? Os outros dois grupos?
Deixa eu ver aqui. Se eu não me engano, um é da Colômbia e o outro é do México. Deixa eu conferir. Mas acho que é isso.
Eu vou ficar aqui quietinha mesmo, aí caso tenha alguma necessidade aí eu posso ajudar também.
Tá bem. Now we're waiting for the team. They're not here yet.
Yeah, they still have 4 minutes, so.
Yeah.
The essential have been taken care of. A new cup of coffee.
They're here now. I'm going to let them in.
No.
Hi, everyone.
Hi.
Hello, good evening.
Hi.
Hey, Cass.
Okay, so I'm just letting in the rest of the team. And we can begin. All right, so just to remind you guys, this is CO2's Naturers, and they have 20 minutes to answer your questions. I will let them know when 10 minutes have passed by, as well as when 5 are left. So you can start whenever you want.
Okay, let's stick with tradition and me going first. First, let me say a really nice project, especially with the combination of doing SynBio and combining that with art. I was wondering, when you, let's say you have made your bacteria, or no, not bacteria, your algae or your dinoflagellates, and you actually release them into the environment. Do you think that they might outcompete their natural brethren because they now can fix more carbon than the ones that are there at the moment?
Well, hello, Christian. First of all, thank you for your question. And well, our objective as for now is to modify these microalgae or dinoflagellates, but to be placed in a bioreactor, like in controlled conditions. We're not planning on sending them to the environment because of what you mentioned, these problematics that could happen when they're competing with other species. So as for now, for biosafety issues, this is what we aim, like only for visual purposes or for educational purposes. So, yeah.
Okay, thanks. And maybe if I may ask a follow-up question concerning the art. It's a bit provocative question, maybe, but I'm a scientist. So do you think that art can achieve something that cold, hard scientific facts cannot to sway the public?
Yeah, thank you so much for your question. I actually believe that through science, we can actually, there is a quote that I really love. It's know the science of art, know this art of science, and discover that all is connected. So through art, we can actually have a special
sense. So I think it's the way in which we can have a broader outreach and combine science with science communication and value for art.
Also, sorry, I want to add something. It's about the way it can be functional. So we think about environmental functional art, and some of our artworks that are in the virtual museum are directed to that kind of art, like the egg. I don't know if you saw it, but it acts like a filter. So if you put it in urban areas and with these dinoflagellates, then it will act as a filter for air and everything, and it will cause curiosity in people. So maybe it will like impulse education and everything else.
Yeah, no, I completely agree with you that using art might actually reach people that you will never get otherwise. Thanks.
I guess I'll go next. I agree with Christian. I love this project. I really appreciated the spin on the art, the VR museum, everything. Really cool. My question to you is technical. In your report, you mentioned that you ran some of your DNA parts through codon optimization, which I appreciate, but you also included some regulatory parts in there, like promoters, RBSs. My question to you is, did you verify that the changes to scrub or remove like restriction sites from those parts didn't impact their function or like change nucleotides and like core regulatory elements?
Hi, Luis. Thank you for the question. Yeah, so since this is just like a prototype or like a demo of what we want to have afterwards, we have not, to be like completely honest, we have not done those details or modified those nucleotide sequences. We plan to do that in the future. Right now we're doing like just, I guess not completely basic, but just like the first steps of getting to what we want to have in the future. So we want this to be a collaborative thing. So if we start having more people using our platform and just sharing their parts with us, so we can make it like a community so everyone can use this platform for their microalgae projects, we can start modifying and optimizing those things that will definitely make our projects or other projects in the world just be completely, how do you say, like workable, usable? Yeah.
I appreciate the honesty. That was more of a flag for you guys to just keep in mind in case like you go through your first iteration of designing, building, and then you test it and nothing works. And you're like, wait, what happened? Why doesn't it work? It's like you introduced a SNP in like the top box of your promoter or something, for instance. But.
Yes, right. Also, the plasmids that we used for now are also the ones that have been used before in other projects for microalgae specifically. So that was our base. But if we do want to continue working on more specific project, we would definitely have to do that. So yeah, thank you for the tip. And my colleague Luis also wants to add something.
Yeah, so I just want to add to give some credit to my partner Andrea Leiva, who was involved on the synthetic biology part. We did some work, especially Andrea, with Chlamydomonas. There is more information with Chlamydomonas, and we do some codon optimization. But as we also include dinoflagellates, that's why we select dinoflagellates, because we found that connection with art, and we found out that there is not a lot of work in dinoflagellates. So like my partner said, there is an area of an opportunity on dinoflagellates to do this optimization and codon optimization and find out that we propose really works because it's what we want, that this works, especially with these promising microorganisms that are awesome, and we are fascinated with them, that are dinoflagellates.
Thank you.
I guess I can go next with a question/comment. First of all, I really like your repository idea. I think that is really cool, especially like with microalgae starting to create, like all databases for most organisms started like that, right? So I think it was really ambitious and a really good take. I wanted to know, like, if you could talk to us a little more about how you envision like your modeling to improve once you start like getting more actual data. Like right now you have like rough estimates, but what have you considered for like future iterations of that modeling platform?
Okay, thanks a lot for the question. That's actually an important core of our project. And well, it's the iterative process that we are intending to develop is the fact that through our interviews and our conversations with the different stakeholders in our community, we devised that to start deploying such technologies in bioart or in CO2 capture and so forth. We need to start to accelerating the design part of these ventures or these ambitious projects. So what we are intending for our database or repository to be able to do is to start feeding on the validation and experimentation of the community in these specific organisms to and serve or act as a tool that will help them identify in a more efficient way the metabolic routes they can take advantage of, or the metabolites that they can produce from the different species, and the genetic information that will allow them to adapt them to the different conditions. We find that when we are trying to do these things in practice, we need to deal with a lot of variation in the environment. So important data, not only the synthetic biology core data, but the photoperiods and growth yield that can arouse from these different species. And when you change the different metabolic aspects of them, that's something that also needs to be, that will be available in this repository and will help in speeding up the deployment of such technologies.
Thank you. And just to restate, I think it's a really cool initial idea. So I look forward. It will be really cool if you can continue it in the future.
Yeah,
you have 10 minutes.
Just to add to what my teammate said, yes, all of that, and we envision it as for the users also to be somewhat of inspiration from the IGEM parts list that has all that side from validation and the teams that everyone can upload their information. And yes, to have both sides from an academic side or people who know a lot of about biotechnology, and also for people that do not have so much knowledge, but to be able to use our platform on a how can I say, on a friendly way to get what they want from our databases.
Thank you.
Okay, then I go for another question. I should know this because we are just resequencing Chlamydomonas reinhardii, but is there a genome scale model for that organism available so that you could actually, before implementing your changes, already figure out what happens if you put a carbon collection module in there? Is there anything known? Because if you collect all the data from the IGEM repository, you might actually know whether there's a model available or not.
Yeah, thanks again for the question. Actually, yes, there's already a repository, but only for Chlamydomonas, and there you can find pretty much everything about it, all of the strains, all the promoters, all the terminators, protocols. So that since that's like the most standardized microalgae that there is, there you can find everything for that. So that's how we based, at least for that microalgae, for that species microalgae. I can look at, I don't have like the link right now with me, but I can look it up like while we're asking and answering and put it in the chat if you'd like.
That would be great.
Do we have any other questions or comments?
I guess I can ask just a curiosity question, maybe if we have extra time. So in regards of, I'm not a person who works with microalgae directly, but how much is known about regulatory elements in that system? Like starting from like transcription factors, transcriptomic data, how much of that is that available? And do you foresee that as a limitation if there's not much data available on that regard? And it's mostly curiosity, so.
Okay, so maybe you could just like repeat the last part of the question just to get an idea on regulations and what specific matter are you saying that if there's not such regulations available, would that be like an impediment or such as an obstacle for our project, or what was it?
I mean more on the like how much is known outside from just like genome sequences, like regulatory elements. Is there like more high throughput studies on those organisms? And if there is not, do you see that as a limitation?
Okay. Yeah, for sure. I think that's actually part of where this whole project started developing, since after speaking with researchers and also entrepreneurs that are working with these organisms, we know that most of their projects are based upon personal experience of and they figuring out what they need to do next. So yeah, I wouldn't dare to say that nowhere in the world or internet is there to be found information on such regulations, but for sure it isn't easy to find, and it's not concentrated in such a way that would allow for a more for a bigger scale development on this synthetic biology ecosystem of microalgae. So yeah, that's for sure an obstacle that needs to be sorted out, and it's actually one of the main goals of this project, is just to start finding these regulations and putting them available to people to really focus on the application part of their projects.
Just a heads up, we have 4 minutes.
Okay. I'll just say something real quick as a follow-up. So yeah, while we were looking at all of the parts on NCBI, there were very few that were reviewed. So like most of them were just, they just belonged to one single paper or publication, and then it was never used again. So that really is the objective of this whole project, to just make it all standardized and available and reviewed. We want to validate all of this information so it can be further used by other scientists and projects around the world.
Cool, thank you, team.
Any other questions or comments?
Cool stuff.
Well, thanks.
Thank you very much.
Yeah, definitely.
From our team, thank you a lot for your time, for your curiosity in our project, and well, we're really hyped up about what's coming on next for this project. We really believe in it and its potential to start developing new technologies that we are going to see more in our surroundings and instead of only like imagining them or see them on paper. So the next step is just along the corner. Thanks a lot.
I'll try to remember to put a link to one of my colleagues who actually works with Chlamydomonas in my comments, so that you might have one person more you might want to contact, maybe.
Oh, great. Thank you very much.
Thank you very much.
Thank you, team. Thank you, judges. Now I will invite you to leave the room and watch the rest of the session on YouTube, okay? Thank you so much.
Thank you.
Thank you.
Okay, so we are moving on to our last team of the evening or the afternoon, depending on where you are, which is chaos. So we already have some members here. I just want to ask if it's just you three. Hang on, 3, 1, 2, 3. Yeah, if it's just you three, or will we have someone else from the team? Chaos, did you understand?
Yes, thanks, Christian.
Okay, so can we begin?
Yes, I think the yes.
Okay, so you will have 20 minutes. I'm going to let you know when 10 minutes have passed and then when 5 are left as well. I'll leave the judges to the questions and I'm here if you need me.
Okay.
Okay, continuing with the newfound tradition of me going first. Let me start with saying that your project looks really, really interesting. And if I understood the concept correctly, it's something I've not seen in that or envisioned in that way so far. So to make sure that I understood it correctly, did I get it right that you basically want to make a genetically modified bacillus that then excretes the D-sub protein linked to the VCF2 protein, and that is then basically taken up by the plant cells and protect them against heat and light stress, right?
Yes, that is right. Basically, our objective is to design a circuit and integrate that circuit into a bacterium chassis that will be Bacillus subtilis. And this circuit, how do you say? This circuit is composed of a protein that is called D-sub, damage suppressor protein, that is a protein that we can find naturally in a tardigrade called Rhamnosus varionatus. And this protein have the property that confer resistance to damage for heat and principally by UV rays. And also this stress is present in Solanum tuberosum plants, in potatoes. And our objective is to integrate that chassis in Bacillus, that is an endophilic bacteria of Solanum tuberosum, to confer resistance to the plants for this stress, the heat stress and the UV rays.
Mm-hmm. So that brings me to a follow-up question. The bacillus is not everywhere in the soil, but can basically go everywhere. So what do you envision would happen if it gets to other plants or other organisms? Would this be a problem?
Okay, excellent question. So in the design of the project, we take account that this could be a risk for the environment and for other plants. So for that, we design a circuit with so special characteristics that is two suppressors, cellulose suppressor and lactose suppressor. So with that suppressor, we can't take control about the dissemination of Bacillus subtilis in other areas, specifically in other plants, and we can take control the expression of the protein in Bacillus. So also the bacteria Bacillus subtilis, as you say, is a bacteria that we can find naturally in the soil. And we consider that the Bacillus subtilis don't carry a problem to other plants. Additionally to that, maybe we need to cover a risk assessment to assegurate that the bacteria is not a risk for other plants.
Mm-hmm. Thanks.
Okay, actually, I love the idea of your project. There is a 2015 movie about a scientist who went to space and only survived by planting potato. So what I want to ask you is, have you thought about an application for your idea in space, like on Mars forever, for example, sorry? And if so, what changes would you make for a project in that sense?
Sorry, I don't understand you. Can you repeat the question?
Okay, on other planets, there is the conditions could be like what you are aiming for this potato plants to withstand. So have you thought of an application for your project on Mars, for example, in space? And if so, what changes you would make?
Okay, it's a very good question. So if the circuit works, maybe they can be implemented in other plants, and these plants could be used in other planets, and maybe the circuit can work not only in plants, maybe they could work in other organisms, and yes, they could be used in other ambients more extremes, like Mars. I don't know if I answer your question.
Okay, I can take that. Thank you.
Thanks to you.
I can go next. I also got my brain thinking about Mars, so that's cool. So first of all, I like to say that I really like your report. It's really well put together. I really like the visuals. Whoever did the tardigrade character, I love it. So really cool. I had some questions about your human practices. So I know that you interview several farmer families and did several activities in that regard. Did you also, so I saw mostly you presented your project. So before that, on the actual conception and when you were collecting ideas, did you also engage with some of your and how do you can you give us some example of some ideas that you got from them that were useful for your design?
Daniel, could you give me a hand with the translation of the questions, please?
También la puedo dar en español, ¿sí?
Exacto.
Okay.
Eso lo iba a proponer.
En sus cuando estuvieron contactando a los granjeros y a las diferentes personas, vi muchas entrevistas en las que ya les presentaban su idea. Quería saber, por ejemplo, qué de lo que les dijeron les fue de utilidad para implementarlo en su... Consideran que, o sea, si nos pueden platicar un poquito más de qué cosas obtuvieron de esas conversaciones que fue útil para su diseño al final.
Pues más que todo como esta parte como de la regulación en cuanto al circuito, incluso a la parte legislativa. Al principio no teníamos el conocimiento adecuado de la parte legislativa, sin embargo pudimos consultar incluso a comités de bioética por parte pues, de otras universidades y también con la ayuda pues, de nuestros mentores. Nos pudimos llevar en cuanto a esto, el tema, por ejemplo, digamos, de lo que había mencionado anteriormente Daniel, que era plantearnos cómo regular este tipo de circuito en cuanto pues, a la bacteria, ya que es una bacteria endofítica de plantas. Pudimos plantearnos y también pues preguntarnos cómo hacíamos nosotros para que esta bacteria no digamos por decirlo, si esta llegara a otros cultivos, esta no los afectara. Entonces ahí miramos el tema, por ejemplo, de lo que son estos represores, como lo del tema de la celulosa y la lactosa. No lo teníamos contemplado hasta que tuvimos estas entrevistas.
Okay, sí, perfecto, era lo que estaba buscando. And I can translate for Christian and Joseph. So just really quick so that we don't take more time on questions. I was asking them what they got from their human practice contacts and talking to stakeholders. And they mentioned that some of the takeouts were the learning about legislation and specific policies that they needed to take into consideration for their project implementation. And then another thing is that they hadn't considered some of the control aspects of like how to avoid like their bacteria and their circuit to propagating or like having some effects on other crops outside from what their target is.
You have 10 minutes left.
Okay, thank you, Tim.
I'll ask a question. Love this project as well. Very cool idea. I have a just curiosity question. Is there evidence that just overexpressing the D-sub protein like in some other organism is enough to impart that same protection like in that new organism? We can't hear you, Danielle.
Sorry.
There we go.
That question. Yes, we have evidence about the use of this protein in other cells. Specifically, we review an article about the use of this protein in mammalian cells. So specifically, we don't search or we don't find investigations with plant cells, but it is also a biological system that could be associated with plant cells. Technically, that is a eukaryotic cells. But yes, the paper has investigation in mammalian cells. No sé si de pronto puedo hacer la traducción y de pronto que los compañeros me complementen ahí esta información.
Understood.
Bueno, nos están preguntando si de pronto tenemos algún antecedente o alguna evidencia sobre la utilización de este sistema, de esta proteína en otros sistemas biológicos.
I guess follow-up question.
Ok. En cuanto a este tipo de proteína sub, no se tiene conocimiento pues, según los estudios no se tiene conocimiento en otros sistemas biológicos, pero de acuerdo pues a que esta funciona como una especie de chaperona, muy parecido a, bueno, muy parecido a cómo funcionan las chaperonas en las células eucariotas, y algo Daniel me puede complementar ahí. Contemplamos también el tema de que la planta pues es una célula eucariota, si bien es una célula vegetal, sigue siendo pues una célula eucariota, pero definitivamente no hay estudios en otros organismos donde se haya utilizado este tipo de proteína.
A follow-up question, if we still have time, do we still have time?
Can you translate? or you understand?
Should I do the translation or?
Yes, sorry.
Okay, sounds good.
That would be awesome.
Thank you.
Yeah.
Sorry.
So yeah. So what he was saying is that there's no like straight evidence that the protein is like will work on another organism, but that basically they are working on the preliminary assumption that since it works as an eukaryote chaperone, and plants are still a eukaryote organism, it should work on a similar fashion as it does on the organism.
Sorry about that. Follow-up question. In your system, you're trying to engineer bacteria to then produce this protein that's then fed to the plant. Is there a reason why you didn't think about just going straight to the plant and just engineering potato plants that can produce it themselves?
Yes, the reason is that we don't want to modify direct the plant. We don't create a genetic modified organisms, specifically the plant, Solanum tuberosum. So for that reason is that we create this circuit and integrate it to the Bacillus subtilis.
Does this not count as GMO? Just out of curiosity.
It's a good question.
Yeah.
So if we take account the legislation in Colombia, the GMOs is organisms that had a modification or a characteristic that is not found naturally in these organisms, and based on that, it could be called OGM.
We have 4 minutes.
Thank you.
If I may follow up on that, I think the potato would not be considered to be a GMO because even if you put a protein from another organism on the potato, that's not genetically modifying it. For me, the question is, how does the protein get to every cell in the potato that actually needs it? Because I think the bacillus will be in the soil, and the parts that get sunlight are the leaves. So how do you envision that the protein goes there? Or doesn't it need to? Is it enough if it's in the potato root itself?
Voy a realizar la traducción para que mis compañeros apoyen con esta respuesta. Nos están realizando la pregunta sobre cómo consideramos que esta proteína puede llegar a todas las áreas de la planta, la parte aérea y la parte del tallo y la parte radicular, teniendo presente pues que la bacteria se encuentra en la parte del suelo, en la parte de las raíces. Entonces, como pretendemos pues que esta proteína pueda llegar a todas las células y las pueda proteger.
Pues en cuanto, pues no, la verdad no lo habíamos contemplado como la parte aérea, Creo que es algo que se tendría que hacer incluso ensayos ya a nivel de laboratorio, una optimización del circuito, pero ya a nivel incluso práctico, se podría decir. Pero lo que sí estamos, digamos por así decirlo, de acuerdo, es en cuanto, y si algo mis compañeros también me pueden apoyar en esto, es en cuanto a que es más que todo como que la planta pueda crecer como, o sea, que esta dependa más que todo como de la temperatura del suelo. Me hago entender, es más como desde la raíz, como la bacteria funciona en las raíces y pues esta técnicamente ese es un tubérculo. Entonces, la idea es que la pueda protegerla de la temperatura, pero de la misma temperatura del suelo, que es la que le está dando pues la radiación, o sea, la radiación más que todo en el suelo, no tanto en la parte aérea. Esa parte no la hemos contemplado, pero en un terreno práctico, por supuesto que se debería contemplar. No sé si respondí tu pregunta. Can you help me with
Translation would be really appreciated.
Yeah. So what the team is saying is that they haven't considered as much the like outside part of the plan. That will be something that needs to be evaluated in like future studies. But what they are mostly considering is that the potato plant, like growing in the ground, like it will be affected mostly by the temperature and conditions that are on the soil. So they are hoping that they can like protect the plant by keeping the properties in the soil. Does that sound correct, team?
Yes, thanks.
Okay, thanks.
Thank you all. Your time is up. I want to thank the team first and then the judges. This is the end of our session. I'm going to ask the judges to stay in the room for a little bit more so we can discuss some things. I'm going to thank as well the public that is watching on YouTube. Thank you so much for being here with us. And we'll see you in the awards ceremony. Bye, guys.
Bye, team. Thank you.
Bye. Thank you to all.
Thank you.
Bye-bye.
Nice job, guys.
Bye.